Ariel is a physicist with a strong background in optics and imaging processing methods. He's working in new instrumentation using polarization and structural light to understand different processes at cellular level. Besides of this, he is working with his partner, Dr. Julia Alonso, developing different instruments for multifocus imaging and light shield microscopy. But today, I think he's going to focus his talk about this snapshot polarimetrics and the phase quantitative. So I will like to thank you, Ariel, to join us and giving this talk, and I pass the word to you.
Okay, thanks, Lionel. Are you hearing me okay over there?
I am looking at your presentation, yes.
Yes, it's okay. Well, thanks, Lionel, for the invitation. Maybe this is the least connected with everything you might have seen before during this workshop, but it's why I'm a physicist, as Lionel said, and my background is in another things a bit different from microscopy, but our interest in microscopy is somehow motivated by that previous work. So I'm going to show you part of our previous work, just to, well, okay, for you to see what we are working on and how we are moving from that things to what is connected to microscopy, in particular to polarimetric microscopy and phase recovery and phase quantification. That's what I'm going to talk to you today. This is part of our work that is obviously not connected to the micro scale, but to a macro scale or a middle scale, where we are able to take different points of view of a certain scene, either by a stereo camera, either by some integral imaging approach. We can synthesize those images, refocus, and so we can work with trying to get rid of occlusions that might be useful also in the biological domain. And what we have learned there is that we might have some different information that has some kind of redundancy that might be useful working on the micro scale. So besides this, that is very nice, at least for me, we tried some image processing at real time using optical methods. In this case, that was part of the motivation that has to do with what I'm going to show you later. That is the use of some kind of lenslet array or a group of lenses in which we have a different point spread function for each one related to, in this case, it's something that has nothing to do with microscopy, at least for the best of my knowledge. There is the generalized Hub transform that is meant to find a certain shape out of a binary scene. In this case, we are working with something that has, for example, certain numbers of circles that you can see in this image, and we want to find every circle for every scale. And to do so, we have some array of lenses that have a different shape or a different size of a circle behind them. That's a pupil mask. And we say, okay, we can achieve what we call a full invariant detection. And we say, okay, we can do this at real time. Why don't we try something different with some physical property or different from geometry and try to apply that on a different scale? And we say, okay, polarization is nice. Let's try with polarized light. As you may know, there are different mechanisms in nature to produce polarized light. First of all, when we talk about polarized light, we are usually talking about linearly polarized light, but the generalized state is of elliptically polarized light, and in particular, circularly polarized light. We know that one of the most common mechanisms is absorption. We have a non-polarized source that passes through some Polaroid, and at the end of this process, we have our light with the electric field that is oscillating in a certain direction. That is polarized light. our vectorial field has a particular direction of vibration. And besides absorption, we know that we can obtain polarization by reflection. You have seen recently one of those mechanisms in action, the reflection of light in the case of turf microscopy. And we can also have a polarization by scattering. At the mix of both, we have this nice phenomena that is called the Brewster dark patch. Besides that, that is nice to see, we know that polarized light can reveal us some properties of the materials. In this case, it's a plastic, and we know that plastic, or under some circumstances, is birefringent, and we can observe this material under cross polarizers. We can have both some of these things that we can observe under transmission, and we know that if the Polaroids or the polarizing plates are crossed, there will be a really small amount of light passing through them. But in the case of these materials, the stress when it was made is somehow codified in this pattern that we can observe under polarized light. Okay, besides these man-made materials, we know that we can reveal some properties of other materials under polarized light. And these materials can also be biological materials. We have tissues, and we can observe, for example, in this nice picture, that what we can't see under bright field or common microscopy, transmission microscopy, is revealed under some parameters related to the polarization introduced by the material. Besides that, if we are talking about diagnosis by polarized light, we know that cancer cells have different properties, mechanical properties, that are revealed by polarized light. Among those properties, or the quantities that we need to measure, are two important things. One is called the degree of linear polarization and the angle of polarization. As we told before, we know that the general state of polarized light might be elliptical, but naturally occurring phenomena always, or most of the times, introduce some kind of linear polarization onto a non-polarized source. So we are mostly interested in that, and it's quite easy to measure. How can we obtain this degree of linear polarization and angle of polarization? Well, this is what is called a Stokes polarimetry, and is one of the different forms that you can find in the literature. The most appropriate for this context is in this regard. We need to obtain at least three parameters that are called S0, S1, and S2 that are related to four measurements that we can perform with our system, our optical system, with one polarization, one polarized plaque in front at what we call 0 degree, 45 degrees, 90 degrees, and 135 degrees. By doing so, we can obtain different images, and we can obtain these parameters that we can in turn give us some information about how light is being modified by the material is going through. The first approach to obtain Stokes parameters, we are missing one here, that is S3, that is related to the degree of circular polarization, but we are going to not to talk about that and probably not introduce it in the first approximation. The first form of how can we work with this polarized light is that we can use the usual or traditional approach, that is using our setup and rotate our analyzer according to the different degrees that we need to obtain the Stokes parameters. Another form that is commercially available is to have what is called a array of polarizers in front of each of our pixels of our camera. This is done at electronic level, and we also have some kind of lens in front of each of our pixels in a micro lens array that allows us to gather polarized light and analyze it through this system. Both of these have certain advantages and certain disadvantages. This is always obviously pretty easy. This is commercially available, but we need to interpolate the information between pixels. And in the first case, we are suffering from motion blur in our images. Another way that it might be much cheaper and somehow easy to apply, at least at the lab level, is to use what I'm going to talk to you about in this new kind of or new development, where we can put behind our objective in our microscope or polarization microscope a lenslet array with different polarization mask behind that. This is the work of Juan Chaguno, who is currently pursuing a degree, a master degree in electrical engineering, working in our lab and co-advised by Federico Lecumberri and me. Our work here is to obtain in a single shot, and this is our motivation, because we want to obtain all the parameters at once and at least try to apply our method to something that is moving. And we are, for doing so, we need to perform some real-time processing. So we can obtain in a single shot in the chip or in the sensor of our camera the different images that are going to give rise to the stock parameters. So as we are putting a different polarization mask behind each of our lenslets, we have what we can see here. There is the image with 0 degree in the polarizer, 45, and so. And we need to put these images in a common geometry, that is what is called in image processing a registration process, and then perform these simple calculations. The registration process is somehow maybe the bottleneck or the most delicate process that we need to do so, and we need to find what is called also the common area. We need to put all this information together, have the greatest field of view possible, and operate. And what we obtain here is an image that corresponds to the previous ones, but only taking into account the common area where we can operate. We are using an Olympus 10 times magnification objective, and our lenslet array is with a focal distance of 42 millimeters. So in whole, we have an effective magnification that is reduced from the original magnification of our objective, but we can operate at real time. So somehow we are sacrificing some level of resolution of our system for real time. This is deeply rooted in the physics of our system, because the amount of information is somehow fixed. So we are not using the whole chip, the whole sensor, but we're using some parts, and what we want to gain is real-time operation capability. This is one of our ongoing results. There are no cells here. Sadly, we are just at our first step, but it's somehow nice to see how it's going. This scale is not perfectly correct, but what we can observe is that at a typical angle of our analyzer, we have all the regions through which the light is passing have a degree of polarization that's more or less uniform. That is okay, because we are trying to see what happens with polarized light passing through something that it cannot introduce any change over it. And the angle of polarization is somehow uniform. And besides that, and that's the most interesting result that I show you today, we can operate at real time. This is the case of a video. I hope you can see it well, where we are moving along as the frames are passing. We are moving the analyzer or the polarizer in front of our system. And what you can observe there is that the degree of linear polarization is not modified. That is okay. So we are operating fine because the linear, our linearly polarized light is not being modified. On the other hand, if we take a look at what happens to the angle of polarization, we can see that it's moving as time passes over our video. So that's okay. And it seems that we can operate fine using our microscope. As you have seen, this is not very suitable for working with live material. So we might have some modification, for example, to change the orientation to a regular microscope. But for the time being, I'm quite happy with the results. Besides polarization, we are also interested in obtaining phase information. What you can see here on the right of your screen is a fish keratite moving. I thank Dr. Miguel Rosena for his video. And what you can observe there, and that's how we connect with the beginning of our presentation, is that we are performing a real-time optical operation, because what we are getting here is phase contrast. Phase contrast imaging is a great example of optical operating over an image. But what we are getting here is just a visualization. What if we are interested in having some information about the amount of phase retardation that our specimen is introducing? So we are moving there to phase quantitative recovery or quantitative phase recovery. As you can see here, you know that phase information is encoded somehow in the focus. An in-focus image or a perfect imager is not capable of giving us any useful information about phase. On the other hand, if we defocus our system below or above the focus position, we have some information. One way to obtain a phase quantitative, besides the common interferometric approaches, is to use what is called the transport of intensity. We are physicists, so we prefer to say irradiance, but it's known in the literature as intensity equation that relates the change in the transversal direction of our phase function with the variation of intensity in direction. Yes? So what we can have here, we need to obtain some variation of our intensity. So this is obtained by different focus positions of our system. This is a traditional approach, for example, where we have on focus, under focus, and over focus. And you can see that phase information along with amplitude information are mixed. But transport of intensity equation allows us to work with both and recover both. So it's a complementary method to what you can obtain, for example, with fluorescence. And by a simple modification of our previous setup, we can develop a real-time processing or real-time acquisition of differently focused images. How we can do this? Because, for example, by introducing a plate that moves our focus, yes, and have different focus positions according to the different thickness of the plates that we can put behind each of our lenses. Yes, this has been introduced with certain success in the context, for example, of AR devices where you need to move the focus. So it's feasible that we can introduce the modification of our over our previous setup, and somehow by operating wisely over this derivative and using 4 images to obtain this variation of intensity in the direction of propagation, we can integrate this equation and obtain phase. By obtaining phase, what you can obtain as from this equation is, for example, volumes of cells. And that might be nice because besides having this only visual information, you can have something quantitative from phase. Well, so this is our ongoing work. Thanks to all our funding agencies, and thanks for the organizers of this nice workshop.
Thank you very much, Ariel. Beautiful presentation and very nice work. Well, we are very good in the time, so we will have space for a couple of questions from the audience. Felipe is requesting for open the microphone.
Hi, Ariel. Thank you for your presentation.
Hi.
I was just curious about the GIF that you show with the cell moving around.
Yes.
There is a halo around the nucleus.
Oh,
what is that there?
I'm just physicist, very out of. I can show it you again if you want, and we can talk over.
I'm
Yes.
not talking about the biology. I'm basically asking why do you see that halo around the nucleus?
Oh,
the bright halo.
I'm not really sure why.
It's not your video.
No. It's not my video, but it was taken under phase contrast imaging, and it's only what I want to show you. That is just the visualization of phase. What we want to do is the quantification of phase.
Yeah, it's fine. It's fine. It's.
I think it can provide us some information about how things are evolving in time there, and in particular for cell migration. So I think hope we have something useful to show you next time.
Awesome. Thank you.
Thank you.
Okay, give me a second. I will look at it here. I don't find any other further question. In any case, if you can stay a little bit, Ariel, we will
have
of
course.
Breakout room. So if there
hope it wasn't too crazy for this.
Fantastic. No, come on.
Okay.
Thank you very much. So now I will move to our next speaker. Michelle, I think you can unmute and share your screen meanwhile I introduce you. Michelle Digman is associate professor at the Department of Biomedical Engineering at University of California, Irvine. She is director of the Keck Nanoimaging Lab and co-PI of the Laboratory for Fluorescent Dynamics, as well as associate professor of developmental and cell biology as well in the University of California. Michelle, as I say in the first day, is one of the world experts in all these techniques of advanced fluorescent microscopy and spectroscopy. She is with Enrico, basically the person behind the development of most of the technique that we have covered over the workshop. In particular, I will emphasize RIGS, NMB, part correlation, the application of facial for film, and many more. During my time at LFD, I always be indebted with her because she gave me the chance to work in close with some of her students, and we have published several papers together. I always thank for this because she gave me a very nice chance. And today, I request her if she can talk about this topic because I found very interesting, and it's a technological development that they are doing there in California to use all the development of film and phasors in the autofluorescence of pre-implant embryos to try to tackle the problem of the fertility too. So thank you very much, Michelle, to be with us, and I pass the word to you for your presentation.
Thank you so much, Leonel. First of all, thank you so much for the introduction. And I just want to say to everyone, buenos dias, buenas tardes, y buenas noches to everyone who's listening in. I'm really excited to talk to you about this work that we've been working at the LFD and show you some of the FLIM applications. And FLIM, of course, you've been learning throughout the workshop. So this project actually began as a curiosity in terms of trying to understand if we could actually develop a way to look at fluorescence lifetime fingerprints of the developing embryo. And there's a lot of literature that has been published over the past 50 years excuse me, that has to do with biochemical analysis and looking at the embryo development from the very early stages all the way up through the late developmental stages. And so what is known about the metabolism of embryos during the stages is that they know that from the 2-cell to 8-cell stage embryo, the pyruvate is the major source of nutrient and energy source for respiration. So there's this very nice figure here depicting that the major component is oxidative phosphorylation. But of course, we know that in every single stage, the metabolism can actually switch. And that's what we are looking for, specific markers that told us exactly when this switch would happen. And if we can follow this in real time and real space, we may be able to use the FLIM signature as an index to quantify embryo health. So hexokinase is inhibited during the first stages of the embryo development. And then around the morula stage, there's an increase in glucose uptake. So there's a switch that happens right after the morula stage. If we take a closer look at each of the stages here and our hypothesis, concluding with the biochemical analysis of the stages of the development of embryo, is that we hypothesize that during the early stages, from the first fertilized oocyte all the way up through probably the 4 to 8 cell stage development, we would see an increase in the amount of NADH that is mostly that could actually dictate the autofluorescence of NADH with the free amount of NADH. So could we look at the ratio between free and bound NADH to see the different stages within the embryo development? NADH is an autofluorescent molecule. I'm going to show you the structure in a second, which is a really nice it's a really nice cofactor molecule, because it is actually involved in cellular metabolism. So NADH is nicotinamide adenine dinucleotide. It's fluorescent in the oxidase stage, and then it plays a central role in energy production. And so in the reduced state is fluorescent. In the oxidized form, it is not fluorescent. So that but we're not looking at NAD+. What has been known in the literature regarding metabolism is that it was actually metabolism can be associated with embryo viability, so the health of embryos. And many studies have shown that embryonic development in multiple mammalian species shows that glucose uptake correlates with embryo health. So that was really nice. It was actually one of the forms where it was hypothesized that if you are able to extract the media that embryos were grown in and quantify the amount of glucose that was left in the media and the metabolites that were produced by these embryos, you can actually quantify whether the health of the embryo was good or not. However, the problem with glucose uptake assays is that they're not very reliable given the number of embryos that are in the Petri dish. So all of this has to be carefully quantified and analyzed. So we know that there is a correlation with metabolism and health. And if we talk to a lot of IVF clinics, so this is in vitro fertilization clinics, and we ask them, what is the gold standard for selecting embryos for implantation into the mother? And the idea today, in today's society, where women and partners wait for a very long time before they have children, and sometimes there's an increase in population of infertility across the world, more and more couples are turning into IVF. So this is becoming a very important area and in clinical settings. And so they say that the state of the art is just the human eye. So selecting the embryo by looking at the morphology and looking to see the quantity, the number of cells within the embryo, and looking to see if there are any abnormalities in terms of shape of each one of these cells. So that is really it's a good way. The human eye is very selective. However, if the clinician is not trained, or if they're very tired that day, they could pick the wrong embryo to implant, and there's only one chance for implantation. So we thought if we could first follow the metabolism of the developing embryo using fluorescence lifetime imaging, we might be able to have a signature to select the healthy embryo. So nicotinamide adenine dinucleotide is the fluorescent cofactor that we're actually exciting using 780 nanometers in the embryo. And the lifetime of the bound state of NADH is very long. It can range between 1.4 to about 9 nanoseconds. And in the free state, you could see that these two rings here self-quench, and the lifetime is about 0.4 nanoseconds. So the imaging strategy that we developed was to isolate mouse embryos from pregnant moms and put them under a CO2 temperature environmental chamber, and then measure the fluorescence lifetime of each of the embryos. To minimize exposure of the embryo, we decided to take a Z-stack of the entire embryo and collapse the entire Z-stack into one final image format. So about 50 frames of a Z-stack integration took about 3 minutes to complete the image. So and I will show you some data showing that under these conditions, we did not observe any DNA damage to the developing embryo. So what does the signature of the autofluorescent path look like? And of course, we're only isolating the NADH emission path. And so as you could see here, we're only selecting the emission at 420 to about 500 nanometers. So this is within the blue emission wavelength of light, and we're not looking at FAD. So in this case, what is very unique, first of all, about the signature of the NADH in these totipotent stem cells is the fact that the FLIM signature looks completely different than any other cell that we've ever imaged. As you could see, the lifetime of the signature of all of these embryos here, I'm going to change the lifetime of all of these different embryos are pretty much dominant on this part of the phasor plot. And it took us a long time to understand why the lifetime is very, very different than the lifetime of the metabolic trajectory that has been shown by Chiara Stringari and many other co-workers at the LFD as to the free and bound final lifetime of the signature of the, let's say, the pure species of NADH and the presence or absence of binding. However, what we decided to do instead of figuring out exactly if we're looking at NADH in a particular bound state, we just decided to look at the differences in the signature for each one of these stages. And in fact, what was really exciting is that each one of these stages actually had a very unique signature. So when we take our cursor and highlight all the pixels that belong to the lifetime here in blue, you can see that the 2-cell stage embryo lights up. And this is the data here. This is the raw data from the 2-cell stage embryo. When we take now our cursor and highlight all the pixels that belong to this lifetime signature, and you could see that the cursor slightly moves over to the right. And so you could see here that that highlights the 8-cell stage embryo. Now there is a switch between the 8-cell to Marula stage here, and you can see that the lifetime starts to shift down towards the center of the fluorescence signal or the phasor plot here. And so as the embryo goes into the late blastocyst stage, it actually, we see that the signature is more or less in the center where we see all the rest of the NADH lifetimes that's typical in all of the differentiated cells that we've looked at. So endothelial, epithelial cells, and so forth. And so we actually are able to show that using the phaser FLIM analysis, we see a movement of the fluorescence lifetime in each one of these developmental stages. And what we wanted to understand is this movement towards the center of the plot. So why is the lifetime of the embryo getting longer? And in fact, you could see that there's a tail here that's going to a very, very long lifetime. And it was also in the literature, if we go back to the literature, we were trying to correlate this lifetime with known biochemical species. And what was known in 2005, there's this paper that was written to show that 8-cell stage embryos have actually a huge amount of reactive oxygen species. And then they decrease in the blastocyst stage. So there's definitely a switch in the 8-cell stage to 16-cell stage to the morula stage, where there's a huge production of the ROS production. And ROS, which is the reactive oxygen species, increases the free radicals that actually are important in development. So this actually has to happen for this embryos to continue development. So there's a very good balance between the production of ROS and maintaining the balance so it doesn't go too much overbalance, and then the embryo can actually die if it has too much reactive oxygen species. So if we correlate now each one of these signatures with the literature from the 1960s, and just as a reminder, these are biochemical analysis where all of the embryos at each one of these stages needed to be terminated, whereas in our lifetime data, all of these embryos are live. We could see that in the 2-cell stage embryo, the pyruvate uptake dominates. So that means that free NADH should go down. And so when we look at the 4-cell stage, free NADH will go up. So this shift of the shorter lifetime, we believe correlates with the increased free NADH at the 4-cell stage. Now from 8-cell stage all the way up to blastocyst stage, oxidative phosphorylation dominates. And bound NADH levels should go up. In fact, the bound NADH, the fluorescence lifetime of what we get of the NADH signal actually gets much longer, which would make sense in the oxidative phosphorylation where there is an increase of bound NADH in the embryo. So the lifetime actually correlates with the signatures that were done in the biochemical assays, where we see a long lifetime, a shortening of the lifetime, and then it gets long again. So we are very excited to see that our FLIM signature correlated with the biochemical analysis. So we then concluded that the phaser FLIM can measure metabolism during embryo development in the implantation embryos. So why is this lifetime getting longer and longer and longer during the reactive oxygen formation during oxidative phosphorylation? We believe that has to do with the fact that reactive oxygen species can actually produce oxidation of lipids. And this was shown by Rupsa Datta, who was a student in Enrico's lab. And what they saw, and this is from their paper, is that the long lifetime species here, which is about 7.8 nanoseconds, correlated with oxidative stress. So the cells here are uptaking oleic acid, and upon the take up of oleic acid, there's an increase of oxidative stress causing lipids to actually oxidize. And we see this very long tail from the signature of NADH. This is, in this case here, this is the trajectory of the what we call the metabolic trajectory, which would just mean the difference from free NADH to bound NADH. So it would fall along this line here. However, you see this tail that is predominantly taking over. And if we look at the pixels that are highlighted, this is where the oleic acid is correlating. So we know that the long lifetime species contribution is due to oxidation of lipids. So as we know in the embryos, ROS production is needed for embryo development. In the bovine embryo in the 8-cell stage, the reactive oxygen levels increase and then they decrease by the blastocyst stage. So ROS is really important in this process. So just to correlate the production of ROS, we wanted to get a better, let's say, sense if this is true, what we're seeing with fluorescence lifetime. We can use other fluorescent dyes. DCF-DA is a reactive oxygen species sensor. And so this, the amount of the ROS does not correlate with the intensity of this particular sensor. However, the sensor does tell us if there's positive ROS production here. And so the green is the positive stain at the 2-cell stage compaction, early blastocyst and late blastocyst stage. In fact, we see ROS in each one of these stages. However, the intensity here in the green does not tell us the amount of ROS. So we decided to use the third harmonic generation to look at the amount of lipids that were being produced during the development of the embryo and see if that we can correlate the third harmonic signal with the long life-type species that we were seeing pixel per pixel. And if we could see that, in fact, the long lifetime species signature in our phasor plot correlated with the lipid using third harmonic, then we could at least infer that the increase of reactive oxygen species was indeed oxidizing the lipids during development. So here we see that at the 2-cell stage, there is no, let's say, localization of the third harmonic lipid species with the fluorescence long lifetime species. However, you start to see the correlation in the compaction, early blastocyst and late blastocyst stage. So we were able to discern that, in fact, this lifetime shift was due to the that amount of reactive oxygen species. So to further look at if we can see at various changes in metabolism, we decided to treat embryos with rotenone and antimyosin A, which are oxidative phosphorylation inhibitors. Now, remember that the embryos at this stage are undergoing oxphos, so they have a high bound NADH production, and each one of these is an embryo here. And so my student Ning was able to use rotenone antimyosin A, and we see a shift towards the free NADH lifetime signature indicative of glycolysis. So we indeed were able to manipulate the metabolism to show that the phasor analysis was a very nice way to see these shifts. We can also do the opposite, where we take embryos that are undergoing glycolysis and shift them back to oxphos using 2-deoxyglucose, which is a glycolysis inhibitor. So here now, this was one of the key points that actually allowed us to look at the amount, the lifetimes of the embryos and actually visualize the embryos to see if we could tell if the embryos are healthy and if the signature of the lifetime had changed. So if we look at the 2-cell stage, there's 3 of them here, and they're under 3 different conditions. The first one is a happy media called the casome media, which is highly nutrient additive media. So the lifetime of the casome condition, as you could see, is here. So each one of these points is the embryo that has been incubated on the happy casome media. Flesh holding media is a nutrient lacking media. And you could see the embryo at the 2-cell stage looks fine. However, you can also see that in the lifetime signature, the metabolism has already switched. And under PBS, of course, PBS, we all know it's just phosphate saline solution. And the lifetime, by eye, these embryos look healthy, but already with the lifetime signature, you already see that the lifetime has completely switched from where the healthy embryos are. So this gave us an indication that we can use fluorescent lifetime, and particularly the phaser approach to analyzing this data as a way and a means to identify healthy embryos from unhealthy embryos, even though they looked healthy by eye. So we now developed a pipeline, a strategy for doing this, super ovulating embryos, collecting the embryos from the moms, and then imaging them. The imaging, time-lapse imaging, took about 60 hours. And you could see here from the movie, we're able to track each one of these embryos and then fluorescently use FLIM to identify the ones that went up to the healthy blastocyst stage and the ones that arrested in the morula stage. These were considered unhealthy. So we were able to use a way to quantify which one of these embryos was better followed, and making sure that we didn't, the 2-cell stage embryos you could see here. This is the receiver operating characteristic curve to see if we followed 2-cell embryos, the AUC gave us about 0.73%, which means that the 2-cell embryos were not very sensitive to looking at FLIM. However, the 8-cell stage embryos were very robust, and they gave us very good data analysis at 0.19. Okay, so this is now just finally, I'm going to close here with the looking at the distance analysis, and I'm not sure if this was actually given in lectures before. So you've heard about the distance analysis, and I'm not going to go into details, but even though if the FLIM signature by eye looks exactly like they are matching, I can assure you that there's very slight changes to the distribution of the fluorescence lifetime signature here. These are healthy embryos that are viable, and these are signatures from embryos that did not go to the blastocyst stage. So these developed to the blastocyst stage, these did not, but we took the data from the very beginning, so 8-cell stage, and we were able to use this data as this test set to look at the distance to get an index of whether the embryos were healthy or not. So the distance program can distinguish between healthy and unhealthy group at the 8-cell stage. And we have here, for example, a predicted of unhealthy were 17 and predicted as healthy were 9. So this is an easy way for clinicians to see whether the embryo is healthy and whether the embryo is unhealthy. And we're just going to go to the final set here. Our accuracy rate is about 86%. So if you go to the clinic, the grading for looking at embryo health at the 8-cell stage is about 57% accuracy, which means like just the flip of a coin, you could just say yes or no. But with the FLIM signature, we could see that it's about 86%. Our precision is actually really high, it's 92%, and that's the percent of correct classification with respect to healthy embryos. So we were able to image many, many embryos. We had a total of 156 healthy from an unknown set, and 53 unhealthy from an unknown set from our mouse experiments. So now we can add the distance analysis to finalize and have an index for healthy and unhealthy embryo. And finally, we can show that FLIM does not cause any DNA damage to embryos. We know that 2-cell stage are really sensitive to light, so we didn't want to use 2-cell or 4-cell embryos. But you could see that the development to blastocysts still occur at even 3.5 milliwatts. If we use 10 milliwatts of power, the embryos arrested or died. So the morula and blastocyst stage embryos also showed a very good percent in development. And finally, if any of you have any questions, if we're able to implant those embryos into moms, we did implantation studies where we implanted the embryos that were imaged into pseudo-pregnant moms, and then we showed that whether we imaged them or not, the embryos gave live births just fine. So we didn't induce any temporary problems to the embryo development. So in summary, the mouse pre-implantation embryo is unique lifetime. We see that the 8-cell stage embryo is optimal for looking at the signature of phlegm. And in our future work, we'd like to investigate the physiology reason for the EBI difference. So in other words, for each one of these stages, what biochemically we're really seeing in terms of the NADH, and look for any chromosome abnormality, such as aneuploidy. So can we identify embryos that have a mismatched number of chromosomes? And then I'll just stop there and thank the people who actually did all this work. I'd like to thank my former student, Ning Ma, who defended her thesis last year, and she's now at Stanford, and did the work with our collaborators, Ken Cho, who's in developmental and cell bio, and all the students in his lab, and all the funding, and all the laboratory for fluorescence dynamics, working in the LFD has been fantastic for my group, and it's been great to work with everyone in the LFD. So thank you so much for your time.
Thank you very much, Michelle. Beautiful data, beautiful presentation. I really like this work. I didn't have the chance to see the last part of the talk before. Is fantastic. All that you can produce and for the valuable information that you can give to physician in that sense. So we have already a couple of questions. The first one is from Florencia Ligoin. And she say, give me a second. Yeah, she say, she want to know if you ever try with the oocyte, and if you think that the metabolic of the oocyte can be useful in order to pre-fertilization, right?
Yeah.
And give also a better chance to have a better finally embryo. And I will add also how know about the sperm, right? Because at the end, they are all very metabolic cells, so, and very special cells.
Yeah, great question. We have not looked at oocyte, and I think that was, one of the things that would be fantastic to do, especially because oocytes can be frozen. And so the idea of, defrosting the oocyte, this is a very critical step in IVF, and selecting the proper oocyte is key, I think, also for fertilization. But we have not looked at the signatures of oocytes. Certainly, this is a very big area. I think we've opened many possibilities for many labs to study this using phlegm and signatures of phlegm. I'd like to add that you could probably use this if you don't have phlegm in your lab, but you have access to multiphoton excitation using spectral analysis, which we have not exploited yet. But it certainly has opened a lot of opportunities to look at the sperm, to look at the oocyte, and to use these non-invasive techniques for selecting the best potential embryo and sperm for development.
I'm looking forward to use this, but as we already have discussed about the importance of the cows here in Uruguay, because we have 7 each person. So you can imagine how important this is. So there is a question from Ernesto. Can you unmute him so he maybe can ask by himself?
Okay. Can you hear me? Yeah.
Yes.
Well, thanks a lot for your talk. We are actually starting a project with embryos, ovine embryos. So, when you fertilize the in vitro or in vivo embryos, there is quite a big difference after then the yield of birth, I will say. And even more, when you freeze the fertilized embryos and you defreeze them, this yield goes below 10% sometimes. It's really, so you looked at the problem, and probably I need to read your paper now. I have to. You looked at the problem, the metabolic analysis, looking at NADH and all these things. About cryogenicity, have you thought about problems in the pellucid zone, or after that in the stability of the membrane, or you know, to that held kind of sealed of the whole embryo before being implanted to the mother?
So we have not looked at any membrane characteristics. That's also another big area that can be done, you know, with using these kind of polar solvatochromatic dyes, maybe using Lordan to see if there are any signatures that are different in the embryo membrane structure. But we have not looked at that. I think that I know that other people were kind of looking at the integrity. There's a study done at Stanford University where they looked at, it's called the squishiness of the embryo. So essentially applying pressure and seeing how much pressure the embryo outputs if you add a given amount of pressure. And they see that that the amount of pressure that the embryo it can output, given, you know, once you add force to it, can be an indicator of health. So we know that it could be that membrane integrity is really important for the health of the embryo. They actually, in the paper that they quoted, also saying that the more, if they induce this amount of pressure to the embryo, they actually have better yield in terms of having viable embryos. So it's actually a good thing to put them a little bit under pressure.
Okay, but in your setup, you looked at fertilized embryos. So you looked at embryos that were not cryogenically preserved. So you took them straight out. They were fertilized.
Yes, that's
okay.
True.
But because this is something that can be compared also after, you know, those that are fertilized in vitro, those that were fertilized in vitro and frozen, and after that, those that were fertilized in vivo.
Yes.
That's something that you can have a look.
Yes, yes.
Okay, thanks.
Okay, thank you very much, Michelle, again. We will split now the speaker in the breakout room. We will recombine in more or less, I would say, less than 20 minutes for the final talk of Dave. We are now assigning to the different speakers, and after, well, you can leave the breakout room whenever you want, but if not, we will cancel the breakout room a couple of minutes before the Dave talk.